Thank you very much for the kind introduction. So today I'm going to take you through one of the, I would say, side projects that emerged throughout my postdoc in Kathy Wu's lab. And I'm just going to share a little bit how we got started with this and then what we learned by doing the project. So one of the malignancies I'm very much interested in is acute myeloid leukemia. And most people who treat AML will be aware that it's a really bad disease. And even if you have a so-called favorable risk subtype, you still only have a 50% chance to be alive after about 5 years. So there's still lots of room for improvement. And one of the cornerstones of AML treatment is allogeneic stem cell transplantation. And here you can to see allogeneic stem cell transplantation in a nutshell, where we basically have donor cells in red that are infused into the patient in blue, and then we try to achieve disease control, meaning we try to eliminate the blue hematopoietic cells, and we oftentimes don't get to zero. There's oftentimes some residual disease and some sort of interaction between the disease and the donor cells. And then, unfortunately, quite a few patients will experience immune escape, meaning the leukemia learns how to escape donor control, and then a relapse will take place. And then the question is, are there ways to reinstate a leukemia control? And this is what we call GVL modulation, so graft versus leukemia modulation, where we try to reinstate leukemia control. And this is something that's quite complex because leukemias have a lot of ways to escape donor immunity. And here you can see a few mechanisms that have been described, and I just want to focus on the T cells today. Sorry, I'm just trying to get the pointer, later pointer. Okay, so I'm going to focus on the T cells today, and I'm especially going to focus on CTLA-4, which is a molecule that conveys immune resistance.
Okay, sorry to interrupt here, Dr. Penter. Can you share your screen? Seems like.
Can I do what?
Share your screen. I don't see your slides.
Oh, that's strange. I'm sharing my screen.
I can see the slides.
Oh, you can see that?
Okay, so I'm going to reshare.
I could see it.
So are we back?
Okay, now I can see that.
Okay, good. So, okay, good. So I was in the fortunate situation that I was involved in the scientific by-program of a clinical trial that tried to improve outcomes after allogeneic stem cell transplantation by testing an ipilimumab-based treatment for relapsed AML. So ipilimumab is an blocking antibody for CTLA-4, and the idea is to reinvigorate donor immunity by blocking CTLA-4. And this trial was very nice because it contained a lot of sampling. So all the participants went through multiple blood and bone marrow collections before treatment and throughout treatment. So this was really a very nice opportunity to understand responses and non-responses on this trial and following this therapy. And one of the interesting aspects of this clinical trial was that there was quite a few responders. So we had plenty of opportunity to understand what's going on in the responders using those samples. And at the same time, there was also quite a bit of disappointment here because there was actually very little very few sustained responses. So here you can see the progression-free survival, and most patients actually progressed after about 6 months. There was one exceptional responder on this trial who's been living for many years, but the rest of the cohort had disease progression. So the question was, of course, also what's going on here? What underlies the fast relapses that we saw on this trial? And so basically, to bring this all together, the question was, what are the determinants of response and resistance to this treatment consisting of decitabine and ipilimumab? And like I said, there was a very dense sampling collection done. So you can see the trial schematic, and we had patients after allogeneic stem cell transplantation, but we also had some that were not transplanted who also had relapsed AML, and they all got decitabine and ipilimumab. And there was, you can see the dotted lines, there was a lot of samples collected throughout this trial. And we performed single-cell RNA-seq on these samples. In total, we performed single-cell RNA-seq on 64 samples from 18 patients, and we obtained about 300,000 single-cell profiles. You can see the UMAP on the right there. And I used a normal bone marrow reference to annotate the cell types, and you can see we found the expected cell types that you would normally see in bone marrow. And we first looked into immune cells, and we saw very clear differences between AML and healthy donors. And we also saw differences between post-transplant and transplant-naive patients. So that was very interesting. And then we wondered whether the T cells would be would associate with response. And actually, the most robust finding in this single cell data set was that the ratio of T and NK cells to myeloid cells was more favorable in the responders. Those are the ones in blue compared to the non-responders. And this was a very robust finding, and it actually persisted throughout therapy. And now you might ask, okay, so what's the significance of this? And if you are an experienced leukemia doctor, you might already guess that the myeloid cells are probably the leukemia. Now, the difficulty with, and this is really the starting point for this project I'm presenting to you today, the difficulty with AML single-cell RNA-seq data is that there is almost no way to know what myeloid cell is a normal myeloid cell and what cell is a leukemic cell, because the transcriptional profiles of normal hematopoiesis and leukemia are very, very similar. And on this trial, about half the patients were post-transplant. And for post-transplant patients, you can actually start to approximate what's the leukemia by looking at donor and recipient derived cells. So this is something that you can do in post-transplant samples by looking at expressed single nucleotide polymorphisms. So we use a tool called supercell, and this allows you to say this cell is likely a donor cell and that cell is likely a recipient cell. And if you now assume that a recipient cell with a myeloid phenotype is leukemia, you have a way to of estimating the leukemic cell populations. It's not perfect, but it's already a pretty good approximation. And for 8 of the patients, I did this and utilized this approach to look at normal hematopoiesis and leukemia, and you can see the results in this heat map. So basically, red means normal, and you can see that the responders and non-responders actually had complete absence of normal hematopoiesis in their HSC and LMPP compartment. So the progenitor cells were basically all malignant. But if you look at the more differentiated cells, so monocytic cells and dendritic cells, you can actually see that the responders had some substantial normal hematopoiesis compared to the non-responders. So this tells you already that there is probably a difference in residual normal hematopoiesis that was relevant for response to therapy. And this is an approach that also allows you to track longitudinal, track responses longitudinally. So here you have a patient, AML 1019, who responded to this treatment. You can see how the blast count goes down. And you can see how the GMP compartment actually improves in terms of normal hematopoiesis that comes back, or donor-derived hematopoiesis. At the same time, we find that even at time of response, there is no recovery of normal HSC or LMPP populations. And I think this is probably the reason why we saw these fast progressions, that even though the disease burden was reduced by the treatment, there was no sustained disease control, since the HSC and LMPP compartment continued to be leukemic. And you can also look at this in another way by looking at copy number changes. And in this patient, we have two copy number variations that allow us to also track the disease longitudinally, and we can see a very similar picture. So this was basically a analysis that I performed and finished, but I was really unsatisfied with the fact that I was unable to track leukemic cells directly for patients who were transplant naive and didn't have copy number changes, because the approaches I've just shown to you don't work with those patients. And there's quite a few of them on that trial and in general. So this brings me to the question, how do we solve where's the AML question in single-cell RNA-seq analysis? And here we got interested in long-read sequencing. This is a novel sequencing technology, and Oxford Nanopore Technologies has one platform for long-read sequencing. And the principle is presented here, where you feed a DNA strand through a pore, and as it goes through the pore, it induces differences in an electric current. And as you record those changes in electric current, you can actually detect the DNA sequence. And the beauty of this approach is that you could feed very long molecules through this pore and get actually very long molecules read out. And this is something that we thought would be very interesting for trying to understand what is the leukemia in a given single cell data set. And this is the technology that we use to develop the NanoRanger platform, or NanoRanger pipeline, I should say. And the workflow is presented here. So we basically use tumor samples, could be leukemia, could be anything else. We perform standard 10x 5' single-cell RNA-seq, and then we get an intermediate cDNA library. And we go ahead and continue with the normal Illumina sequencing, which gives us the cell identity that everyone knows from single-cell RNA-seq data sets. But at the same time, we also take a portion of the cDNA and we perform a PCR that allows us to amplify transcripts that contain natural barcodes like somatic mutations. We then sequence those with Oxford Nanopore. The reason being that this way we can cover the entire molecule, and we're not constrained so much as with Illumina sequencing to certain parts of the molecule, and this has a lot of advantages for the analytical part. And we then are able to detect different molecular features such as somatic mutations, but we can also look at other variants with mitochondrial DNA mutations. I'm going to talk about that, or T-cell receptor sequences, etc. And then we integrate that with the Illumina sequencing data, and we get an integrated analysis where we now can say, okay, this or that cluster has this or that mutation, and this allows us now to actually directly identify leukemia cells. I just want to briefly show you the PCR. So it's a 3-step PCR where we first amplify the cDNA further and remove template switch oligo priming artifacts. We then do a first PCR where we amplify a transcript of interest, and then a second nested PCR to increase specificity. And we then go for sequencing, and then we also have developed a pipeline that actually allows you to process that data. So anyone who knows something about Oxford nanopore sequencing is going to be surprised, and they're going to ask, okay, does this really work? I remember that Oxford nanopore sequencing doesn't have a great sequencing accuracy. Can you convince me that this actually works? So I want to give you an example that hopefully will convince you that this can work. So here we took a tumor sample from a melanoma patient, and we performed TCR, T-cell receptor sequencing, using the 10x platform. And this is something that is, an established protocol, of course. And we did the standard Illumina sequencing, where you get fragmented parts of the TCR, and then you have to assemble that bioinformatically, and this is how you get T-cell receptor information. And then we actually took a portion of that DNA and we sequenced it with Oxford nanopore sequencing and analyzed it with our workflow and compared the data that you get out of this between Illumina and Oxford nanopore sequencing. And the reason why we chose TCR is that this is a very delicate molecule where every nucleotide counts, and you really care about the accuracy of the part of the TCR that recognizes an antigen. And if we compare the Oxford nanopore with the Illumina single-cell sequencing TCR data, we actually see very good alignment. So here on the left, you can see the number of raw reads per cell. And then if you now look at the CDR3s, so the part of the TCR that recognizes an antigen, you see there's almost perfect alignment. So this tells you that the quality of the Oxford nanopore data is actually sufficient to identify genetic variants that are quite delicate, such as TCR. And this is just to say that we find basically the same results. So let's try to track responses to immunotherapy. Let's go back to the clinical trial that I introduced you to in the beginning. So this is the UMAP I already showed you of the single cell data that I previously analyzed. And now at the bottom, you can see the same UMAP, and you can see the cells that have a mutation. So black means they don't have a mutation, and red means there is a somatic mutation. And you can already appreciate that most somatic mutations actually fall in this myeloid compartment and also in the megakaryocytic and erythroid compartment. So this is kind of expected for AML. There's also a few red dots, for example, in the NK compartment, NK cell compartment. I'll get back to that in a moment. So one of the first things I wanted to do is to convince myself that the somatic mutation detection is valid. And I compared the supercell approach, where you define donor and recipient cells to approximate leukemia, with the NanoRanger approach. And you can see on the left identification of donor and recipient cells, and on the right you can see the identification of somatic mutations, and there's actually very good agreement. So this was something that was quite reassuring, that we find somatic mutations in the expected cell compartments, and they align with other ways of identifying leukemia. Now, do we see changes in the leukemic compartment throughout therapy? So here on the left, you can see a UMAP of cells from patients before they entered the clinical trial and received treatment. And on the right-hand side, you can see a UMAP of cells from patients at time of response. And you will probably see very quickly that there is not a lot of change there. So if we do this in a more statistical way, you can actually see that the mutation burden across 5 different cell compartments are actually very similar. So this is further evidence that the treatment on this clinical trial basically reduced disease burden, but didn't really clear leukemic cells from the progenitor compartment. So this confirms the previous analysis that I had done and shows how you can use this approach to track responses to therapy. Now the question is, does decitabine and ipilimumab only lead to this cytoreduction, or is there more? And I want to share one example with you where I could actually see more than just cytoreduction. So this is one patient, AML 8007, and you can now see the cells at screening, so prior to therapy. We're looking at 651 cells. At the top, you can see their genetic profiles. We can see 3 somatic mutations. We can see 3 chromosomal aberrations, and we can really see how we have a block of leukemic cells, and we can also see that they express the expected surface markers, and they also have the transcriptional profiles of leukemic cells. For example, they all express CD34. Then we also have the immune cell compartment that, of course, is absent of somatic mutations. Now I'm going to show you the same heat map for cells at time of response. And now we can actually see that the treatment does induce changes. So if we focus on the 2 chromosomal changes at the top, amplification of 1p and deletion of 3p, we can actually see that in some of the cells, these go away, while the somatic mutations, they actually persist. So this is an example where we can actually see differential response to this therapy. And if you put this all together, you can imagine that we have a, more resistant founder clone of this leukemia that has deletion 5q and the 3 somatic mutations that persist at time of response. And then you have a subclone that's defined by these other 2 copy number changes that has a higher sensitivity to this treatment, actually partially goes away at time of response. So this is just to say that if we look very closely with these approaches, we can actually find therapeutic effects beyond just cytoreduction. So I already mentioned that we also find somatic mutations in non-leukemic cells, such as immune cell populations, and I want to take you through that a little bit more. So here you can see the mutation burden, if you will, for each cell compartment. And again, on the left-hand side, we have the leukemic cells. So we have myelopoiesis, megakaryopoiesis, and erythropoiesis. They all have lots of mutations. So that's AML, that's expected. But if we now look at the immune cells, we see that NK cells seem to stand out. So most T cells don't have any somatic mutations, but in the NK cell compartment, we do see somatic mutations. And this is, of course, interesting. The question, of course, is what's the reason for this? And I think the most probable explanation is that this is due to the fact that most of these patients had secondary AML after clonal hematopoiesis, and the NK cells are probably also derived from expanded stem cell clones that precede leukemia, so pre-leukemic clones that give rise to NK cells, and that's why the NK cell compartment also has somatic mutations. And this is something that can actually be relevant for tracking response to therapy. So here I want to show you clinical data from a patient that was treated with different treatments over time. And to track response to therapy, clinical bulk amplicon sequencing was performed, where you basically take DNA from the bone marrow and then you do targeted sequencing of known mutations. And then by looking at the variant allele frequency, you can actually say whether the leukemia, recedes or comes back. So it's it's a pretty nice way of tracking leukemic burden. And in this patient, we see an interesting pattern. So we have two sets of mutations. We have the red mutations that go up and down and up. And then we have two TP53 mutations that do the opposite. So as the red mutations go up, they go down, and vice versa. And you might wonder, by looking at this data, do we have two competing leukemia clones, or what's going on here? And if you now perform single-cell RNA-seq with the approach that I'm presenting to you, you can actually understand what's going on here. So if we look at the two TP53 mutations, we see that they're basically only found in NK cells, while the other mutations are found in the leukemic compartment. And with this information, you can now understand this much better. So this the two TP53 mutations don't, they're not a marker of a competing leukemia clone. They're just NK cells that become more detectable as the leukemia goes into a remission, or the disease burden drops, because then when you do bulk sequencing, you pick up more DNA from NK cells. So I think this is actually a very nice illustration how the single-cell genomics field could actually improve current diagnostics for tracking therapy responses in leukemia and probably also other malignancies. Now I want to talk a little bit about mitochondrial DNA mutations. This is something you might have heard about. It's something that has gained a lot of interest in recent years. The basic principle here is that mitochondrial DNA is very abundant. Each cell has hundreds of mitochondria. Each mitochondrion has its own genome, and the mutation rate is 10 times higher compared to nuclear DNA. And since the mitochondrial genome is fairly small, we can actually capture it at single-cell resolution. So this is a very nice way of performing lineage tracing in single cells. And in the post-transplant context, there is an additional layer here that's very interesting. If we look at donor cells and recipient cells, they actually have their own sets of mitochondrial mutations because they have mitochondria from different sources. So if you look at mitochondrial DNA mutations, you actually have a way of distinguishing donor and recipient cells. And if you now look at mutations, mitochondrial DNA mutations that are leukemia specific, and you put it all together, you actually have a way of tracking leukemia relapse after transplant very precisely. And I just want to show to you that this works quite well. So here we have a heat map with cells from a bone marrow from a patient who relapsed after transplant. And you can very easily see that we have 4 mutations that are recipient specific and 4 mutations that are donor specific. So they very nicely distinguish donor and recipient cells. And if we now focus on the recipient cells again, we actually see that in the red colors, the leukemic cells have additional mitochondrial DNA mutations that are absent from the immune cells. So this is a very high resolution way of tracking different cell populations after transplant. And I just want to convince you that the donor-recipient deconvolution with mitochondrial DNA mutations is pretty good by comparing it with donor and recipient identification using single nucleotide polymorphisms. And we actually see very good agreement with both approaches. Now, how do mitochondrial DNA mutations align with somatic mutations? Here I'm showing you recipient cells from a patient who relapsed after transplant, and we're looking at mitochondrial DNA mutations and somatic mutations. And I think it's pretty clear that the alignment is very high. So this is it's nice because the mitochondrial DNA mutations actually give you similar insights into the leukemia as somatic mutations. And at the same time, the information is not the same. There are some nuances. And I just want to show you one example from a different publication. So this is a bit of a different story, but it still is a very nice illustration how mitochondrial DNA mutations and somatic mutations actually are quite complementary. So here you see an AML, again, relapse after stem cell transplantation, and we're looking at different cells. Each column is one cell, and we can see their protein expression at the top, and then in the middle we have somatic mutations and mitochondrial DNA mutations at the bottom, and we can see how there's very nice alignment between somatic and mitochondrial DNA mutations. But actually, in fact, if we just look at the cells that I labeled main clone 1 on the left, we can see that these cells can be subdivided further using mitochondrial DNA mutations. So this is an approach that actually gives you further insights into the leukemia. And by reading out both barcodes, you can really you can understand the subclonal structure very nicely. Okay, I just want to give you one more example how these two approaches complement each other quite nicely. So here I'm showing you a diagnostic sample from an AML that was supposed to have an NPM1 mutation. And when I used NanoRanger to understand this leukemia a bit better, I actually realized that there was 2 different NPM1 mutations that were just 3 nucleotides apart. So one codon difference, but nevertheless detectable. And you can see the UMAP on the left, or the UMAPs on the left, and the purple cells are the ones that have one of the 2 NPM1 mutations, and the green cells are the ones that have a different NPM1 mutation. And you can also see at the bottom row, you can see 4 different mitochondrial DNA mutations, and 2 of them actually co-segregate with the purple cells, and 2 of them co-segregate with the green cells. And if we look at this in a heat map, we can really see the mutually exclusive subclones in this case. So this is an AML that actually has very clearly identifiable subclones, and they're defined by 2 different NPM1 mutations, which is quite curious. And since NanoRanger also gives you transcriptional profiles, we can now ask, okay, so what's the difference in gene expression between the 2 subclones? And that's shown at the bottom. Okay, now long-read sequencing, of course, also gives you the ability to look at isoform expression. And the question was, since we have the possibility to look at long-read sequencing data with NanoRanger, can we find isoforms that are relevant for ipilimumab-based therapy? And one example is CD45. So people who work with T cells probably know that CD45 has different isoforms, such as CD45RA and RO, and they can be used to define T cell subsets. And by performing targeted sequencing of the PTPRC gene, which encodes for CD45, you can actually distinguish these isoforms by looking at exon 4. And we did this, and the nice thing is that if you perform targeted sequencing of the PTPRC gene, you can actually dramatically increase the coverage of cells where you pick up PTPRC transcripts. So in this knee plot, you can see if you just perform long-read sequencing on the whole transcriptome amplified cDNA, you only capture a few cells. But if you now do targeted sequencing using NanoRanger, you can actually get thousands of cells and look at their CD45 expression. And this actually works remarkably well. So on this plot on the right-hand side, we can see the expression of exon 4 of PTPRC. So that's basically CD45RA. And I'm showing you the expression in different T-cell subsets. For each subset, we see the expression before ipilimumab therapy, and then after 1 and after 4 cycles of ipilimumab. And you can see how the CD45RA expression actually goes down. And we see a very similar picture if we look at protein expression. So this is site-seq data, same samples, and you can see that the CD45RA expression actually also goes down. So this is actually a known effect of ipilimumab, which leads to T-cell differentiation. And it was very nice to see that you can pick up this effect even without looking at protein expression. What about CTLA-4 itself? So this is actually also an interesting transcript. CTLA-4 comes in 2 flavors. You have soluble and membranous CTLA-4 defined by the differential expression of exon 3. Again, if you do targeted sequencing with NanoRanger, you can actually dramatically increase the number of cells where you have information on the expression. And what I did was to compare the expression of the membranous CTLA-4 isoform across AML and other T cells, we can actually see that there are quite dramatic differences in the expression of membranous CTLA-4 in T cells in AML compared to T cells in melanoma or even CAR T cells. So this is another interesting aspect that you can pick up with this technology. I just want to now, before I come to an end, talk a little bit about how good is targeted genotyping with NanoRanger. This is something that's probably an important limitation and something that, of course, you have to be aware of when you do these experiments. So here we mixed 2 different AML cell lines that both had a TP53 mutation, and we looked at the TP53 mutations in the mix. And if we find one or the other TP53 mutation, we have a way of distinguishing these two cell lines. And what we found is that no matter at what ratio you mix these cells, you actually have a pretty stable genotyping rate for these cell lines. And it was interesting to see that one of the 2 cell lines actually had a pretty low genotyping rate of about 2%, while the other had a 20% genotyping rate. And we did find the TP53 mutations in these expected cells. So the genotyping works well, irrespective of cell fractions, but there are quite important differences in genotyping rates. And one could, propose to just sequence more, and we tried that. And there's actually a pretty quick saturation when you increase the sequencing depth with long-read sequencing. So the genotyping rate is not really determined by your sequencing depth. It's something else. And one clue to what this else is came from our attempts to look at fusion transcripts. So we tried to look at BCR-ABL and RUNX1T1, or also called AML1-ETO. Those are very, very long transcripts, and we actually had a very poor genotyping efficiency for these fusion transcripts. And as I thought about this, I actually realized what the problem is and what the limitation of targeted genotyping of cDNA libraries is, namely that the representation of the molecules in the cDNA is very much dependent on their length. So if you have a short transcript length, you actually have a very high representation, and the longer the transcript gets, the lower the representation in the cDNA. On the right-hand side, you can see a heat map where you can find the same effect. If you have a very short transcript, you actually get coverage of the entire thing. If you have a longer transcript, at some point you start to only get either the 5' or the 3' end, and you are unable to get any coverage from the middle of the transcript. And if you're trying to genotype something that's very long, you can actually only get a small portion at the 5' end. So I think this is an important limitation of NanoRanger. And I just want to show you one more piece of data where we actually try to improve the genotyping efficiency by combining it with an approach called GOT that was developed by Dan Lander, where you actually spike in primers during the RT step in order to improve the coverage of transcripts that bear a mutation of interest. And we did this, and this is able to improve the genotyping rate a little bit, not dramatically, but this is one option to actually improve things a little bit. And what was interesting is that the combination of primer spike in with long read sequencing was actually most effective, and that's because if you do Illumina sequencing, you can actually only sequence molecules up to a length of about 1.5 kb. If you want to sequence longer transcripts, you need to use Oxford Nanopore Sequence or Sing or any other long read sequencing platform. So with this, I want to summarize. So I hope I've convinced you that NanoRanger is a very versatile protocol for targeted genotyping of 5' single-cell RNA-seq libraries. We could also do 3' if we wanted. We would just have to change the primers a little bit. And this allows you to do combined tracking of genetic and transcriptional profiles, for example, to understand therapy responses. It improves the detection of alternative splicing. And one important limitation is that the genotyping rate is really dependent on the transcription levels of your target, the distance to the 5' end, and the transcript length. And primer spiking can improve this in some cases. So lots of people involved in this project, and thank you for listening.
All right, thank you so much for the great presentation. Do we have any question from our audience? If you have question, please raise your hand, and I think Dr. Kinney and I will try to unmute you, and you can also type the question in the Q&A session. Okay, there's one question from Ashish. Go ahead.
Hey, hi. Thank you for the great talk. I do have one question regarding the pipeline. In the pipeline, you actually mentioned that like a part of the cDNA and DNA, you actually take it to do the sequencing through your pipeline. Can you tell us like how much do we require for that, for the sequencing, the cDNA and DNA for your pipeline? NanoRanger.
So the question is, how much cDNA do we need for this? When you elute the cDNA in the 10x protocol, you alluded in a volume of like 30 or 40 microliters, and you need about 10 for doing Illumina sequencing. So you have about 60 to 70% left. And out of that, we typically used 5 microliters. So, 15% or something of the cDNA. And with that, you can do all the sequencing you want. So it's not a lot of material that you need, and it's comparable to what you need for performing BCR, TCR sequencing.
Okay. Thank you.
Do you have any more question?
I have a question. So I think like thank you for the great talk. I think it is actually probably very difficult work, like a lot of things with and I'm just wondering like how did you do the cell type annotation? Like because those progenitor cell types, they might be quite similar. I'm just wondering like how certain you are with the annotation with those progenitor cells.
Yeah, that's a very good question. I mean, cell type annotations is something that you can really spend a lot of time on. And I think one of the really efficient approaches nowadays is to use reference data sets where you perform a label transfer. So this is what I've done. I've taken a annotated normal bone marrow and I performed label transfer, and then this basically automatically defines your cell types. I think you always need to double check that, so don't just blindly accept that, because there's caveats there, and different reference data sets give you different labels. But I think this is a pretty convincing way of doing it, and when you check the marker gene expression, you actually find that they make a lot of sense. So that's how I did it. I used label transfer and then verified that the cell populations had the expected gene expression profiles.
Thank you.
I have one question. Great talk. That's sort of more of a big picture question. So what is why is it that in some of the cells you're seeing so many different types of mutations as well as mitochondrial mutations in leukemia? Do you have a theory? Can you explain it with one concept, why you see that? I know in cancers, they accumulate mutations over time, but particularly the adults, for example. But I just don't know enough about leukemia and sort of the process. And is it the same as like solid tumors, or is it different?
Yeah, it's a very interesting question. I think so it depends a little bit on what mutation you're looking at. So mitochondrial DNA mutations are typically something that happens during cell division. So it's like a DNA replication error usually. So if you have a cell that replicates a lot, a malignant cell, then you really have a chance to accumulate a lot of mitochondrial DNA mutations just due to the number of cell divisions they went through. With the somatic mutations, I think you need to distinguish, driver mutations and passenger mutations. And I think what's very interesting is to look at non-recurrent mutations, because there's many more of those compared to driver mutations, and they can tell you a lot about the history of a leukemia. And I think those are also something that is induced by this DNA replication error. Not so much, genotoxic effects. I mean, leukemia is not lung cancer or something that's due to exposure of the environment. If you look at the mutational signatures, it's actually mostly cell, DNA replication errors.
Right. Thank you.
All right. Seems like we don't have more question from our researchers. Well, maybe the other question, maybe last question I have is, so for the nanos, for the nano ranger, you know, I think it's very interesting tool. Do you have any like future plan for this tools? Are you going to have like a nanopore nano range version 2 or any other future plan you have for this software?
Yeah, absolutely. So one aspect I'm thinking about is how to scale this up. So in this work, I've looked only at like 3, 4, 5 mutations per case. I'm thinking about how to increase the throughput to be able to look at 50 mutations in a leukemia. This is something that requires a bit of change in workflow. So that's something I'm working on. And then in terms of like what's the actual application beyond just a technical toy, I think monitoring leukemia would be something I'm very interested in using single cell genomics to understand, for example, what happens when you have residual leukemia after therapy. This is something that is pretty inaccessible so far. You can do flow cytometry, but that doesn't really tell you much about resistance mechanisms and so on. So I think this approach, it has a lot of potential to be utilized for understanding residual leukemia or longitudinal changes in leukemic cells through therapy. And this is something I want to explore with this tool.
Great. Yeah, looking forward to the updates in the future.
Okay.
All right. Well, thank you so much for the great talk, and we really appreciate that. I hope all of you have a great day.
Yeah, thank you.